Phototransduction takes place in the retina where light is converted into a neural impulse. This occurs mainly in photoreceptor cells, the rods and cones. Exposure of retina to light hyperpolarizes the rods, but when light is absent, rod cells are depolarized. There are components called rhodopsin inside rod cells, which are composed of opsin and retinal, which absorb light. In the dark, the retina will be in the cis-11 state. As soon as light hits the retina, it causes it to change to 11 transretinal. This change of configuration generates an event that triggers opsin to activate the transducin protein. The transducin then activates the protein phosphodiesterase. The phosphodiesterase starts breaking down cyclic GMP into regular GMP. This causes a decrease in the concentration of cyclic GMP. This leads to the closing of sodium channels and results in hyperpolarization.
Let's talk about the ion channels in the dark and light. In the dark, the ion channels are open. The influx of the positively charged sodium and calcium ions has a depolarizing effect on the cell. The inward current is opposed by positively charged potassium ions following out through potassium channels known as hyperpolarizing effect. The combined result of the movement of ions cause the depolarization of the photoreceptors in the dark. In the dark, the depolarized state of the membrane, which is at approximately -40 mV, triggers continual transmitter release, more specifically, the glutamate from the synaptic terminals of the photoreceptor cells. Absorption of light reduces the concentration of cyclic GMP in the outer segment, leading to the closure of cyclic GMP gated channels. As a result, positive charge carried by potassium ions flows out of the cell more rapidly than positive charge carried by sodium and calcium ions flows in. The cell becomes hyperpolarized, that is, more negative, and decreases its release of the neurotransmitter, namely glutamate from the terminal.
And now we're going to look further into the pathway of signals in the visual system. To detect the appearance and disappearance of light, the retina has on and off channels which are present at many levels of the visual pathway. And the first layer to have these channels is at the bipolar cells. In the outer plexiform layer, the on-center bipolar cells make an invaginating synapse with the photoreceptors, while the off-center bipolar cells make a flat synapse. On-center bipolar cells signal light increments, and off-center bipolar cells signal light decrements. Glutamate is a neurotransmitter released by the photoreceptors which has different effects on the two types of bipolar cells. During light stimulation, less glutamate is released. And for on-center bipolar cells, glutamate is inhibitory. When there is a reduction in its release, excitation of the bipolar cells takes place. This is known as depolarization. For off-center bipolar cells, glutamate has the opposite effect, meaning that it is excitatory. When there is a decrease in glutamate release, inhibition of the bipolar cells takes place, and the cell is hyperpolarized. Bipolar cells transmit graded potentials to ganglion cells, which is the second layer to have on and off channels. Bipolar cells that connect only to cones have both on and off variants, whereas bipolar cells that connect to rods have only the on variant. Rod bipolar cells pass the signal to ganglion cells via A2 macron cells. Some A2 macron cells make connection to on-cone bipolar cells and others to off-cone bipolar cells.
Now let's look at midget and diffuse bipolar cells. We're going to look at what these cells are, and the differences between the two types of cells. Midget and diffuse bipolar cells are types of bipolar cells which we can find in the structure of the retina. Both of them are formed by cone centers and work as a transmitter that transmits the chromatic signals in the retina. Midget bipolar cells are cells that have a small soma and its receptive field center receives input from one cone only. That is why it can only receive one signal and transmits that signal to the ganglion cell. It also has less dendritic trees than diffuse bipolar cells. Additionally, it is divided into two types, which are on-midget bipolar cells and off-midget bipolar cells. Both of them connect with on and off ganglion cells respectively to transmit the signal. Diffuse bipolar cells have a large soma and their receptive field can input more than one photoreceptor, and it's formed from 5 to 10 cones. It also has more extensive dendritic trees. To recap this video, we spoke about the action of ion channels in the dark and light, phototransduction, on and off channels in the visual pathway, and finally midget and diffuse bipolar cells.